Okay, welcome to this session entitled Hematological Factors and Aerobic Performance, the Role of the Blood. This is part of a wider series on endurance physiology and physiological profiling for endurance, but also can be viewed as a standalone session on its own right. Now, the purpose of this session is fundamentally threefold. We're going to start by examining the composition of the blood and then the structures that are involved in oxygen transport. So that's kind of just giving us a sense of the anatomy of the blood itself, not of the arteries or the veins, but fundamentally of the blood itself. Then we're going to think about how blood volume and concentrations are affected predominantly by aerobic exercise. And we're going to look at that under different conditions. So for example, what happens if we remove blood, as in we do a blood donation? What happens in response to training, and what happens, for example, to these blood markers if we expose somebody to a condition such as altitude. And then, of course, overwrapping all of this, we're going to determine norm values for these blood-based parameters. Now, the reason we're focusing on these hematological factors, if we're considering a wider journey, which is that of endurance physiology, is that in essence the whole process, which we can see on our screen here, which is this idea of moving and using oxygen. So here's the oxygen being consumed. This is at the mouth, so VO2, volume of oxygen at the mouth, the amount of carbon dioxide being exhaled at the mouth. The ability to move that oxygen to the point where it is needed, in this case, which is in the mitochondrion, where in essence we have the QO2, which really reflects the oxygen consumption at the cell, and the QCO2 reflecting the carbon dioxide production at the cell. That process of moving the oxygen to the cell and the CO2 away from the cell is dependent on the blood. Fundamentally, the transport of oxygen through the respiratory system into the cardiovascular system and then into the localized vascular system is only facilitated in the presence of having the blood, primarily the red blood cell containing the hemoglobin molecule that allows for the transport of the oxygen to and from the cell. So understanding the role in which the blood plays is very important. It also helps us to understand if we're trying to explain why we have got, for example, improvements, or why we've got negative responses in some of our key endurance parameters. It might simply be just referring to what's happening in terms of the blood itself and the blood as a variable, as to kind of explaining, well, why is it we're seeing the deficits in these responses? So we'll start quite simply by thinking about the composition of the blood. And in this kind of crude animation, if we spin a blood sample, we take a whole blood sample and spin it, this is theoretically what you would end up with in a healthy population group. So 55% in a healthy population group of the whole blood is plasma, the aqueous composition of the blood, and it has that kind of slightly yellowy tint to it that we see when we spin the blood down. You've got the vast majority, which is less than the plasma volume generally, which is the erythrocyte, fundamentally the red blood cell. And then you've got the platelets, which is normally less than 1% of the whole blood. And then we have, sitting between those, we have what is called the buffy layer. And the buffy layer fundamentally is the demarcator between what is in essence the plasma volume and the erythrocyte. So what happens is if we were to increase the erythrocyte concentration, so we increase it from say 45 to 50%, in the same volume of blood, the only thing that can allow for that is we would have to fundamentally reduce the plasma volume. And that is where we change the viscosity of the blood. The blood fundamentally becomes thicker. And that has ramifications, as we'll deal with as we get further into this. That has ramifications in terms of the ability to move the blood, and it changes the blood pressure, because if the blood becomes thicker, it becomes harder to move. Blood pressure has to increase to compensate for that. The same if we were to, for example, get a reduction in erythrocyte count, the red blood cell count. Fundamentally that is achieved, and what tends to happen is, as that volume falls, there's a requisite increase in the plasma volume, because this is fundamentally taken across normally 100 ml sample. So these are kind of 55%, so about 55 mils of 100 ml sample is plasma. 45 mils of 100 ml sample are erythrocytes. So when we think about the blood cells themselves, we're kind of looking at the constituent of the blood cells. What we have to think about is that process. The process is really engendered from the notion of erythropoiesis. And erythropoiesis is the process by which red blood cells are stimulated and by the process by which they are produced. And erythropoiesis is stimulated by the presence of erythropoietin in the blood. And what stimulates that? Well, predominantly the primary stimulant for that is carbon dioxide. And if we get an increase in CO2 in the circulation, that stimulates the process of erythropoiesis, which triggers the formation of producing red blood cells. Now, you can probably imagine where there might be examples of that being highly stimulated. So, for example, were we to increase the CO2 concentration either in inspired air or simply by exposing the cell to raise CO2 during training, for example, both of those would over time trigger the process of erythropoiesis. So as you'll see as we get further into this session, exposing somebody to altitude, where although we don't change the atmospheric concentration of CO2, what we do is decrease the partial pressure of oxygen in the atmospheric air. And by decreasing the partial pressure of oxygen, we increase the PCO2 in the cell, because fundamentally you're reducing the PO2, you're reducing the amount of oxygen that is coming in to the cell. The amount of CO2 that is left at the cell increases. That will over time stimulate the process of erythropoiesis and red blood cell production. It is akin to what we see with training. If we expose somebody to training above particular exercise intensities, that is kind of where we're starting to push the cell into an environment where it is producing an excess of CO2, we'll do a similar thing. It will trigger erythropoiesis, but the magnitude of the erythropoiesis will not be to the same scale as we would see when we expose something to these conditions at altitude. When we think about the hemoglobin molecule, and of course what you've got to remember is the hemoglobin molecule sits on the red blood cell. The hemoglobin molecule is actually a fascinating structure. Well, it can combine with both oxygen and carbon dioxide. So what you end up with is oxyhemoglobin when you've got O2 bound to the hemoglobin, and then you've got carboxyhemoglobin excuse me, when you've got the CO2 bound to it. But what is fascinating is actually the hemoglobin molecule would actually much rather combine to CO2. Its affinity is about 250 times greater than it is for O2. Why? Well, fundamentally, it comes back to the fact that CO2 creates a noxious environment in the cell. It creates the environment in the cell which triggers anaerobiosis. It starts to trigger or lead to potentially things like decreases in cell pH, all of which over time can potentially be quite debilitating to the cell. So the key is that we actually want to remove the CO2, and that's why the hemoglobin actually has a greater affinity for carrying CO2 than it will do O2. So when we kind of look at the structure, so we take this rider here, if we've got our red blood cells, so this is kind of a standard structure of a red blood cell. They're a unique structure in the sense that they're highly pliable cells, that they change their shape. So each corpuscle changes its shape under pressure. So although you have this kind of spherical structure that you see here with that kind of natural indent that you see, actually, depending on the pressure, they can change their shape. They become more elongated as the pressure in the cell. So as you start to get a vasoconstriction, the cell actually compresses and it becomes almost oval shaped, and actually quite narrow oval shaped, to allow it to move through the compressed vascular system that we get under the vasoconstriction. When we get the dilation, the cells then readjust their shape to what you kind of see here. And then what we have sitting on the cell are millions and millions of the hemoglobin molecules. And you can see here we've got a very nice kind of example of the hemoglobin molecule. So what you have here is we have our kind of natural protein structure, and then sitting in this you have the hemes, and then you have the iron molecules. And what you can end up is you've got these four structures here, which refers to the fact that we can combine to 4 molecules of oxygen can combine to each molecule of hemoglobin. So you fundamentally have the heme structure, which is this kind of network. Then you have the globin molecules, and the globin molecules contain an ion, and it's the ion combined with the globin that creates the environment for the attachment for the O2 or the CO2 molecules.